In days gone by, intrepid explorers set sail into the unknown, embarking on perilous forays that uncovered new lands and charted unfamiliar territories. 20th century exploration, too, plunges into unmapped regions, mysterious areas beyond the reach of the microscope. One such voyage of discovery was launched here in Cambridge, England in the 1950s, when Professor Sir John Kendrew first succeeded in using a technique called X-ray crystallography to reveal the three-dimensional structure of the protein, a discovery that soon after earned him the Nobel Prize.
Proteins and nucleic acids are the two most exciting and important kinds of molecule that you find in living organisms. And proteins are very complicated molecules with thousands of atoms in them. And of course, biochemists would like to know how proteins work, because it is the proteins which manage all the chemical reactions going on in living cells.
More than 3 decades later, Sir John's pioneering research is the inspiration for a project initiated by a team at Israel's Weizmann Institute of Science, which sets out to find a crucial protein, an enzyme called acetylcholinesterase, involved in many brain and neurological disorders. The quest begins in the Mediterranean with a fish called the electric ray torpedo. This strange creature is an excellent source of the elusive acetylcholinesterase. This enzyme serves a vital function in our bodies. Nerve impulses are transmitted across junctions by a very small molecule called acetylcholine. In order to clear the channels for a new impulse, the enzyme acetylcholinesterase goes into action, breaking down the acetylcholine molecules at the phenomenal rate of 20,000 molecules per second. In the lab, the acetylcholinesterase extract from the torpedo fish is carefully processed. In a multidisciplinary effort, a group headed by Weizmann neurobiologist Silman has succeeded in isolating and purifying the acetylcholinesterase to an exceptional degree, a difficult and painstaking task. This very pure material is then crystallized. The crystal is now ready for examination in this X-ray crystallography lab, one of the best equipped of its kind in the world. The ultimate result of the X-ray analysis depends to a large extent on the quality of the acetylcholinesterase crystal. A crystal is carefully mounted and positioned on the tip of this needle. The X-ray electronic photography produces an array of spots called a diffraction pattern, which is a coded image of the enzyme structure and is the basis for the subsequent analysis. At this stage, state-of-the-art high-speed computers come into play. Institute structural chemist Joel Sussman and his team converted the coded information into a contour map, which gradually revealed the 3D structure of the entire enzyme. Like the unexpected continents that loomed before explorers of old, these modern-day mapmakers were confronted with a surprisingly deep gorge at the heart of the molecule. The scientists have subsequently shown that this cavity leads into the active site, where the acetylcholinesterase manages to break down and destroy the neurotransmitter acetylcholine, shown here by a Y-shaped symbol. The walls of the cavity provide numerous sites to which the potential medications for brain and neurological disorders can bind.
I think it will make it possible to design drugs which will be similar but not identical with this molecule acetylcholine, and which will be of value in regulating the brain function and coping with those diseases.
The day when a pill for forgetfulness will be available over the counter is still a long way off, but the Weizmann research does open the way for the development of possible treatments to inhibit acetylcholinesterase activity and thus alleviate the acetylcholine deficiency, characteristic of Alzheimer's and other all too prevalent diseases.